Some people have already suspected it, but no one really wanted to believe. Scatter-based nanoparticle tracking analysis is obsolete. What does it mean for us? Do we really have to say goodbye to NTA forever? Hi, my name is Sascha Raschke. I'm application specialist at Particle-Metrix, and yes, this is a somewhat provocative statement. So let's take a teeny tiny moment to provide some clarity. Scatter-based NTA allows very quick and reliable determination of size and concentration of all particles in your EV preparation. But the big limitation is that deeper characterization of EV subpopulations in your sample is not possible. I'm sure I'm not telling you anything new here. Fluorescence-based NTA, however, makes use of highly specific fluorescently labeled antibodies or other suitable dyes to allow for much more detailed results, because you can phenotype your EVs highly specifically. So far, so good. But what if you could determine 2 or more specific fluorescence markers simultaneously in your EVs in one single colocalization measurement? And what if you didn't have to do without precise size and concentration measurements in the individual fluorescence channels? Here are 5 questions and answers about colocalization NTA to help you improve and optimize your EV research. At the end of the video, you will have a better understanding of how colocalization NTA works, how you can plan your experiments, and I will give you a few tips and tricks on how to establish the fluorescence staining for colocalization NTA.
What is the benefit of colocalization NTA?
Colocalization measurement allows visualization of 2 or more different fluorescent labels, each with a separate emission wavelength, to determine whether the different targets are in the same region. Colocalization NTA takes place in liquid, which means that Brownian motion can still be captured. So one benefit is that in addition to colocalization detections, you will still be provided with size distribution and concentration information of your individual fluorescent subpopulations in only one single measurement. In addition, you can perform colocalization NTA with commercially available primary antibodies, even in combination with specific membrane dyes. The market is huge, and special developments are definitely not required. Since colocalization NTA happens in liquid, there is no time-consuming immobilization or fixation of the particles, there are no slides, and less pre-experimental work is required. What else? Very little sample is necessary. In many cases, only 1 microliter of the original purified EVs is sufficient for complete fluorescent labeling. And last but not least, since there are so many antibodies and dyes on the market, there is no need for expensive ELISA-like kits or any other kind of click chemistry.
How does colocalization NTA work?
Colocalization measurement means visualization of 2 or more different fluorescent labels in a biological sample, ideally in one single run. With colocalization NTA, the same fluorescently labeled particles are illuminated very quickly, one after the other, with 2 or more lasers, and detected in the corresponding fluorescence channels. In this example, the labeled particles are excited with the blue 488 nanometer laser and after that with the red 640 nanometer laser and detected in the corresponding fluorescence channels. Since the Brownian motion is also tracked, the switchover takes place very quickly. As a result, you get a representation of the XY centroid coordinates of the fluorescence scattering events of the particles. It consists of information about your EVs showing a signal in the green and a signal in the red fluorescence channel. Double positive particles are highlighted by a blue cross because they represent colocalization events.
What to consider for good colocalization results?
Basically, there are some general challenges that need to be addressed to get good results. For example, the user-to-user variability needs to be minimized. In an internal lab study, we examined the influence of the experience level of users on the repeatability of NTA results. 3 users were asked to stain EVs with a fluorescently labeled antibody. Each staining preparation was split into individual batches, and was diluted and measured multiple times on the ZetaView instrument. The results of user A shows a large variation, and you see that the results become more consistent with the further experience of user B and user C. What does it tell us? Training and experience of the user is an important factor of the experimental output. Another key point to pay attention to: optimizing dye concentration or low signal-to-noise ratio. What does it mean? Staining with fluorescence means adding dye in excess, which in turn means high amount of unbound dye. Ideally, you wash all free dye or antibody away, but you are working with very small particles, and compared to much larger cells, they have a very limited number of antibody binding sites. So washing would cost us a lot of time and we would lose a big portion of particles of interest. The solution is to test several dilutions of the dye or antibodies. Optimization should be done in a way that only the labeled particles can be seen in the live view in sufficient numbers, and the concentration of unbound dye or antibodies is below the detection limit of the ZetaView instrument. That means, in most cases, pre-dilution of the dye or antibody for the labeling reaction is highly recommended. Optimization does not only refer to the adaptation of the antibody-vesicle ratio, but also in general to antibody concentration, incubation temperature, and also incubation time. Last but not least, choice of fluorophores. When using a combination of different fluorophores, carryover from one to the other fluorescence channel needs to be taken into account, and like in flow cytometry, the spillover effect needs to be corrected. The best way, however, to avoid spillover is to use combinations of fluorophores whose Stokes shifts are far enough apart. Furthermore, you should prefer bright and stable fluorophores. The keyword here is photobleaching. And no, it's nothing to be afraid of. Photobleaching has to be taken into account wherever you work with fluorescence, not only with colocalization NTA. The great thing about the ZetaView instrument is that you can immediately see photobleaching in the live view, even without having to start a measurement. When doing multistaining, you should preferably choose combinations of photostable fluorophores. And what if you plan a colocalization measurement and you might want to combine fluorophores that have different photostable properties? The ZetaView is highly flexible, and you can adjust the instrument in that way that the fluorophore with less photostability is excited first, and then immediately measure the more photostable dye. This may reduce bleaching of unstable fluorophores. So based on the dye combination you use for phenotyping your EVs, you can tell the instrument exactly in which sequence the fluorophores should be excited and measured.
What staining strategy is recommended?
In our experience, it is advisable to establish the appropriate single stainings first. All you need to do is a little experiment in which you test the right dilution for each dye or antibody to set the signal-to-noise ratio optimally. And by observing the live view, the ZetaView instrument can help you distinguish labeled EVs from the background. So what comes next? Once you have established the individual single stainings of your EVs, you can continue with a double or multi-staining. For this purpose, you can try to combine the protocols of the individual single stainings. And, very important, note that the concentration ratios between EVs and the dyes that you found in the single stainings should be the same in the multi-stainings. What about the labeling sequence? The more dyes or antibody you use for colocalization NTA, the more comprehensive the order in which the different dyes can be added. Do my colocalization ratios change with different labeling sequence? We tried it once and stained MSC EVs with anti-CD9 Pacific Blue, anti-CD41 Alexa Fluor 488 and the membrane dye Cell Mask Deep Red in a different order. And as for this kind of multistaining, we could not find any significant difference in colocalization results for different staining sequences.
And what about controls?
No controls, no reliable data. Controls are important and they have their place. I'm not telling you anything new when I say that the ISEV guidelines describe requirements and experimental controls which must be conducted for a proper EV analysis. One important control, which also applies to colocalization NTA, is a detergent control after staining. In our example, the non-ionic detergent NP40 disrupts the vesicle membrane. No membrane, no surface molecules in close proximity, no colocalization. And that's exactly what we can show with colocalization NTA. So this test shows that we are dealing with EVs. For another check, you put everything into the labeling reaction, but instead of the EVs, you put the appropriate volume of buffer. No EVs, no labeling, no colocalization events. Check. And to wrap things up, FMO controls. They are originally derived from flow cytometry. During labeling, you omit one dye, but still test for colocalization in all available fluorescence channels. As a result, depending on the combination of the dyes, you get only one set of fluorescence channels in which you can detect colocalization. Taken together, colocalization NTA offers the ability to quickly detect the expression of different surface markers using multiple lasers and flexibly selectable pairs of fluorescence filters, including the benefit of sizing and concentration measurement of fluorescent subpopulations. And everything is combined in one single instrument. Colocalization experiments certainly require a bit of time and fiddling, but we are sure that if you follow a tip or two, you will get very good, accurate and reproducible colocalization results. You can find more details in one of our latest application notes on our website. I say goodbye, thank you very much for your interest, and I wish you all the best and good luck for your experiments.